I'm a director here at Open Pharma Research, and on behalf of Open Pharma, I would like to welcome you today for our next digital dialogue sessions. Today's topic is generating AI and ML ready data in drug discovery and development, the strategy and the execution. We have a great panel of speakers today who are going to be sharing with you their some of their work and their insights. I'd like to thank ZONTAL for their support and input in the development of today's agenda. And before we go into the meat, what I'd like to do is to go through a little bit of housekeeping. First of all, is to let you know that this digital dialogue session is being recorded and will be made publicly available at some point in the next few days. We also would encourage you to ask questions, and you can use the Q&A function on the Big Marker platform in order to do that. So what I would suggest you do is, if you have a question, just place that in the box and press carriage return. That will get to us, and we'll save those questions for the panel discussion, which will follow the presentations. The other thing that we're going to do is to run a series of polls over the course of the session today, and that's going to help us give a bit of backdrop and context to the discussion. So I hope very much that you'll be able to take some time to complete those questions for us. And then at the end of the webinar, we'll have a slide of some key contacts within Open Pharma Research for you to contact should you wish to follow up on any of the topics that we covered today. And we'll also have some information on the next activity. So welcome again to our panel, and I think you can see in the slide in front of you the panelists that we have taking part today. So Matthew Woodcock, welcome, Matthew. Matthew is head of data science and infrastructure and tools at Tools, Data Science and AI at AstraZeneca. And then we have Vijay Raju, who is Global Head of Analytical R&D for Novartis, and Wolfgang Kolsmann, who is CEO for ZONTAL. So welcome, gentlemen. And what I'm going to do now is to start the first poll. So can I ask Luke to run the first poll for us? So you can see it here there in front of you. What are the biggest barriers to generating AI and ML ready data? So if you could let us know what are the biggest obstacles you're facing in generating AI and ML ready data. So if you could spend a few seconds pondering that and then submitting your answer to us. Okay. So perhaps no surprise there. The biggest barrier is almost equal in importance, are disconnected data sets at 40% and lack of standards at 38%. So thank you for completing that poll. And what I'd like to do now is to start with our first presentation. So Matthew, welcome again, and the floor is yours.
Thank you, Zahid, and welcome everybody. I'd like to spend a few minutes telling you about AstraZeneca's Science Data Foundation. And as you can see there, the tagline is Analytics Ready Data for Machine Learning and AI. And I'll explain how we came to that. Interesting to see the results of the poll. These are all things that we recognize, including skills and training, that are barriers to machine learning and AI. So hopefully you'll see during the course of the next few slides some of the ways in which we're hoping to address those. So if we think about how we operate today, and this slide's a couple of years old now, so the less than 2 years is where we should be now. We tend to think about analyzing data within individual data types, whether it's omics or commercial data, interactive media, imaging, clinical data. And we recognize that we get a lot more value and we can answer more complex questions through being able to bring those data types together. And that's been done historically through point solutions and the ability to integrate specific data sets together. What we'd like to try and move to is something which is closer to what you might call algorithmic intelligence, where we're trying to bring together data sets on the fly because we've ingested them, mapped them to standards, and made them available to be able to cut and slice through the data as required based on the scientific question you want to ask. So opportunities to extract scientific insights using data science and AI are across the R&D pipeline. We've started our efforts in the early phases around new targets and new leads, and that's partly because of cultural issues where we wanted to embark on being able to use particularly AI in an area where there was still a lot more of validation and traditional pipeline work that needed to be done to get to the end point. Interestingly, the push for us now from our senior stakeholders is how can we use these approaches in late stage pipeline impact? So we've seen something of a pivot over the last 2 years. Broadly, what we're trying to do by bringing all this data together and enabling it to be used for machine learning and AI is to enhance the speed of projects, but also enhance the specificity of projects. And the argument about starting with target biology is the better your target, the easier your clinical trial is going to be downstream. This is actually a slide we used with our senior executive team, so that's the CEO and his direct reports, in 2018, and this was the result of some strategic work that had been done that year, bringing together all the different concepts that we're now building. And so this was about being able to bring together data sources across multiple data types, internally and externally, building out core platforms that we would use for that data management. We're ingesting all of the data into a lake in the cloud, and then we have a variety of ways of organizing that data, whether it's data marts, knowledge graphs, or even a data warehouse. And the idea is that we can use that to support a variety of advanced AI-based analytics. So thinking specifically about the Science Data Foundation, when we created the strategic program, it's fair to say the interest from our senior stakeholders was around the exciting machine learning and AI and what it was going to do for them. And then what we had to reveal is that to do that, you need good quality underlying data, data standards, metadata models, pipelines, and the ability to bring data together, both in terms of the data integration, but also in terms of how you pipe that into analytics environments. So we have an awful lot of data, as you might expect for any large pharmaceutical company, including we have 23,000 clinical studies in varying quality levels of annotation and data completeness. We've got a large-scale genomics strategy, which we announced in 2016. And in addition to the data that we sequence from our own clinical patients, we also have the UK Biobank data with all of the whole exome data that's been provided, along with a number of other pharma companies and consortium. And we're increasingly finding that imaging data and real-world evidence data are also vitally important. However, and I think this is probably true for a lot of scientific companies, we've got a long history of generating data to answer the immediate question. So we generate the data to answer the null hypothesis, whether the experiment's at the bench or the bedside. What we hadn't previously done is considered how we were going to reuse that data to answer more complex questions. So we want to be able to not only generate that data to answer the specific question, but generate that data with reuse in mind. And there's logic to this in that as we're thinking about developing new drugs, we want to make sure that we are as close to our target organism, i.e. us, as possible. And the more we can reuse data generated from observing drugs on humans, the more likely our future drugs are to be effective. So the idea is, and this is a concept that I'm sure you're familiar with now, is that we want our data to be fair, findable, accessible, interoperable, and reusable. And I think along with a lot of other people, initially the focus is around the F. So we're looking to make sure that our data is findable. And again, in a company the size of AstraZeneca, with 13,000 people in R&D and a long history of experimentation, that data isn't always as findable as you'd like it to be. So we've had a long focus on data catalogs, dataset descriptions, study level metadata, being able to pull together the information that we need. That data needs to be accessible, and that goes in two ways. So we took a decision about 3 years ago now that we would make our completed trial data available for research purposes down to the individual patient level, provided there was a good research reason to do so. And that's fantastic, but it brings with it a certain amount of obligations in terms of security and compliance. So we are now able to get down to individual patient level data cross trial, but we need to make sure that we do that in a secure way. So what we're doing using the data lake that I mentioned earlier on is we're bringing all that data together in one place. We've also got lots of data in other locations. So we're looking at how we can use virtualized graph technology to integrate that data. And then potentially we use what I call federated iterative querying. So you would query the clinical data in one location, pass through a set of cases versus controls, if you like, depending on the criteria you've used to select the patient level data, to a genomics environment, carry out some genomics experiments based on the sub-selection that you've already made, pass back the genomic information into your clinical data, and iterate through like that. We want to be able to ask complex questions of our data, so the data needs to be interoperable. And there's a number of ways in which that's important. So knowing that you've got common IDs between your data sets seems trivial, but is actually an important part of being able to bring data together. So patient ID, study ID, sample ID are all vitally important. If you don't have that data in combination, you can still look at gene level integration or some other types of integration. But within data types, so for example, looking at omics data, how do you know if your data is comparable? So we need to think about understanding the experimental design. So in addition to producing analytics ready data for machine learning and AI, we're also thinking about machine readable experimental design, machine readable consents, machine readable clinical study protocols, so we can understand the derived values and how they're calculated. And this is all driving towards R at the end, which is the reuse, which is where we'll extract the value. So thinking about the Science Data Foundation, what is it? It's a collaborative program. We're doing this together with our IT colleagues, very tightly intertwined. We've put a lot of effort and investment into building out platform capabilities in IT to enable us to do this. It's an enduring capability. We're not running a project, and when the project is finished, everyone disperses to the four winds of other sides of the portfolio and does something else. We're standing up teams that will look after this because we recognize this is an enduring thing. We're not going to stop generating data when we finish calculating the relationships between them. It's going to continue. We're going to create new data types. We're going to have new ways of integrating the data. So we need to do this for the foreseeable. It's a change management program enabled by some technology. It's not actually a technology program. So it's about encouraging data capture and tagging for reuse. And some of this is cultural. We need to change the way in which people record the metadata against their data. And nobody likes to record additional metadata, more than 2 extra fields, and the compliance level drops off. So we're not only looking at peer opinion in terms of is your data reusable by the next team downstream, we're also thinking about the reward culture in the organization as a whole. Do you get rewarded for the completeness of your metadata on your data set, for example? What it isn't is a one-time effort, and what it isn't, despite many people wanting it to be, is a cleanup effort across every single data point in R&D. Lots of people would like us to go in and clean up their data. This is about providing analytics-ready data for machine learning and AI. It's not just the data, it's also the pipelines, the processing, the compute, and it's associated with compute environments to enable you to do advanced computing. So essentially containerized areas on the cloud where you can pipe the data in that you've received access to through your request process, and a set of tools that will enable you to do your work. Right now that's expert level, but we're aiming to introduce this kind of capability to what we call the citizen data scientist downstream. And this is we did actually put this in the senior executive deck as well, although on reflection, it's a little complicated. The idea is to show that what we're bringing together is data from multiple different data sources, all the way through from omics and imaging data, but also chemistry reaction data, structure activity relationships and analytics. But then also there's a big emphasis around the individual and study level patient data, real world evidence, and bringing in data from other external sources as well. Some of that data we will ingest, some of that data we'll query in situ, depending on the rights that we have to that data as part of our licensing. But if we've got large amounts of data sitting somewhere else, we don't need to bring it in-house to be able to query it. We can query it in situ. And then the idea is that we enable people to sub-select that data to support their machine learning and AI. Now, I mentioned earlier on that we had individual patient level data as part of this solution, which is fantastic, but comes with a certain amount of obligation. So we're also validating the environments. We didn't set out to validate the environment, and some parts of the environment are essentially unvalidatable. So you've got the interesting challenge of having a hybrid environment. And the way we're managing that is it's all based on what's the purpose of the activity. So if your endpoint is an exploratory enhancement of your understanding of the biology behind a particular disease, and you're going to use that to drive novel targets, then you don't need to be operating in a validated manner. If your endpoint is a change in the protocol for the next iteration of the study, or a model that you're going to apply to patient stratification, or something that has a validated endpoint, then you need to operate in a validated manner. So we've taken the decision to validate the data storage environment, the lake, and the analytics environments, what we're calling AI bench. The naming competition for that was long and complicated, and so that you will be able to support downstream pipeline impact, not just the early leads and targets that I was talking about at the beginning. But to do so, we've built compliance into the whole process, which is an overhead, but it enables us to drive more valuable application of this kind of approaches to the whole of the R&D pipeline. And you can see there we've got what we call the intent to insight process, and I'm going to touch on that in a little bit more detail. But the idea is that you ingest these data into an environment that allows you to then sub-select them, run them through analytics, and get actionable results out at the end. So this is the general goal around generating analytics-ready data. So we're bringing the data in. Right now, we're working through a backlog, but over time, we're working with the core capabilities that generate the data on behalf of the company, whether it's a core capability in the lab that's generating omics data, or a CRO working on our behalf, generating imaging data as part of a clinical trial. We've got ingestion pipelines that we've automated that bring the data into the lake. We can also bring in data from a variety of lab instruments. The coverage for that so far has been around small molecules, but we have additional systems which we use for bringing in large molecule screening and assay data. And I think the challenge there is reusability, particularly in the large molecule space. The number of assays that you run, you may only run them a few times, whereas in small molecules, you run them hundreds of times as part of your design, make, test, analyze cycle. Then once the data is ingested, we automatically add that to the data catalog, and also auto-enrich the data by comparing it to our reference data management system. And then we can take a look at that data and map it to the AstraZeneca standard, see how compliant it is with the standard, what's the completeness of the metadata model, how conformant is it to a range of synonyms that are acceptable for that type of data, so we can see what quality of data we're looking at. Additionally, and this has been something which caused a great deal of debate, we do have the ability for people to bring their own data to the environment, but we've got quality stamps on whether that data is something that somebody wants to use inside their own project, something which they've brought in that's reusable for a variety of projects, but comes with a caveat, comes with a warning that it's not the master copy or high quality data, or data that we've copied in from an authoritative source. This data is reusable in a validated way, and you can use it to support a validated endpoint, for example. And then I'm not going to dwell particularly on the analytics environments, but what we're building is, once you've requested the data, then it will automatically get piped into a standalone analytics environment for the people who've got the rights to access that individual patient level data and any other associated data that ties in with it. And the reason for this is partly GDPR regulation. Because we've got individual patient level data, we need to think about GDPR. And one of the tenets of GDPR is that you should only access the data for the duration of the reason why you're accessing the data. So once that activity is complete and there's a set of results that have been generated, we extract those results from the container, remove the data from the container, and then archive off the container. And in doing so, not only are we attempting to verify the data, we're attempting to verify the analytics. So we've got complex logging on the data in the environment. And the idea is that you would be able to unarchive that environment, be able to identify the data that was used in the analytics, and if necessary, reproduce that analytics. And the logging that you have in the environment essentially acts as your audit trail. So last couple of slides. So I mentioned seamless intent to insight. So you've got to put in a number of different capabilities to support this. You need to have a powerful and intuitive interface to be able to find your data. And actually, we've realized that you need to find both the data that's available in the lake and the data that we have that isn't in the lake, so that people can request request ingestion, so the data can be put in the right environment. Then once they've made that request, right now that's a manual process. So there are people with seasoned compliance experience looking at requests, and we've had about 300 requests to access clinical data through this process so far. It's going up every day. We've got probably about 600,000 patients worth of data in the system now. We're aiming to get to 1 million by the end of the year. Sometimes that data volume goes down if we sell products. If we divest something, as it's called, then we have to take the data out of the environment. But there's a continual ratchet to bring that data in. Then the data is provisioned into an analytics environment, and then that can be used for performing training a machine learning model, or performing other types of AI. And then the information is extracted to support decisions downstream. And I've mentioned most of the pieces around compliance by default. This is we started when we started the project, we weren't expecting to be needing to be compliant, but we realized quite quickly that we would. And I'm glad we did, because as I mentioned at the beginning of my talk, the pivot has happened in the organization where it's not just focused on early stage. The confidence in our ability to use machine learning and AI in complex decisions is such that they now want to apply that to the late stage pipeline. And anybody who's tried to validate a complex hybrid system like this will realize how much effort has to go into it. So I'm glad we started a year ago, otherwise we'd be a year behind. So what we're aiming for is new target biology, AI-driven lead optimization, and as I mentioned, we are now looking at late-stage pipeline impact through the reuse of this data. And we have 1,000 studies at the end of 2019 and 1 million patients in 2020, along with imaging, omics, and lab analytical data to try and drive those questions. Okay, that was all I had. So happy to take questions in the panel session at the end.
Wonderful. Thank you very much, Matthew. And as Matthew has mentioned, we will be saving questions for the panel discussion, which will come after the next session. So do keep the questions coming in. And what I'm going to do now is introduce you to our next speaker, or rather two speakers. And we have Vijay Raju, Global Head of Analytical R&D for Novartis, and Wolfgang Kolzmann, CEO for ZONTAL. And they're going to take us through through enabling the digital lab, data science, and data strategy. So, gentlemen, the floor is yours. And I think we're going to begin with Vijay.
Great. Thank you, Zahid. So overall, as an organization, Novartis has made data and digital a big priority, and this cuts across all our business units, be it research, development, or in the commercial part. I want to talk to you a little bit more in the context of the CMC organization, which is hosted within development, but of course, we also support our partners on the research side. Our aspiration is to be an industry-leading CMC organization, but specifically also to ensure that we have our colleagues who are digitally savvy and are able to use such data and digital technologies. And the reason this is important, I think you also saw that in the poll, it is not only important to have these capabilities, but also to have the skills and training for the associates who are able to use them, because at the end of the day, they need to own and engage with these things. Now, what I want to do is to go through a few examples in terms of how we are enabling this. So one persona I want to share is in the area of technical development. Here is Sabine. So what this colleague usually would do is throughout the life cycle, starting in the early stage, come up with a drug substance and then develop a drug product, do the analytics associated with it, and then do experimentation and do more analysis, more results, and then keep doing that throughout the entire life cycle. So it is quite possible by the time Sabine goes from early stage to the time of handing over to our commercial production, maybe there is 20 variants of drug substance, maybe 30 variants of drug product. And for each of them, there is, again, a lot of our experience from the past. What we want to now do is move away from that, move more in the direction of being a digital first mindset. What that would mean is to enable more and more digital twins as a starting point. So for example, if someone needs to do crystallization experiment, can we actually do a modeling and simulation to understand those behaviors? Similarly, if you're doing a process development with a roller compaction or a granulation, to be able to do modeling and simulation. So more we are able to do of that, we believe that will allow us to reduce the number of experiments, potentially also use more of the historical information. And once we generate all the data, we are also able to use process automation to put the data into reports and things like that, with the intent that all of this on one hand is able to reduce the time frame, maybe from years, potentially also to shave off a few months at a minimum, if not more, but also allow our associates to then to refocus on science and do something that they really excel at. Who is a technical project leader. In her world, she needs to keep an oversight of the overall development in terms of all the reports we're generating and all the knowledge we are generating. One of the challenges that she has today is, again, like Matthew said, we have experience with maybe 50 or 100, if not more, products just in the last 10 years. And sometimes we have exactly the same kinds of problems, maybe slightly different ones in a different project. However, it is extremely difficult for us to figure out what issue happened previously, what we did with it, who was the expert involved in it. So what we have been able to do now is, again, in partnership with with some of our external partners, move more into the area of data science, where we are doing more and more cognitive search. We are also able to do things around similarity inference and document summary. So the beauty of these things is, imagine you just have a Google-like interface. You have a certain keyword you type in. It gives you potentially what alternate options exist for that keyword. And then when you select the right one, it is able to provide a deep link into an existing document. So with a few clicks, you're already able to look at all the historical information. And we've seen that it has a huge potential in terms of reducing the amount of time and frustration for our colleagues, and really enabling them again to use past information in a more seamless way. I then want to use another persona here, and here is Matej. Matej is working in a biologics lab. One of the complexities with large molecules is, if you look at a chromatogram, it is fairly complex. You may have sometimes 20, 30 fakingly, manually aligned, assigned, and then integrated. And the amount of time it takes, and when you have to do this for thousands of samples and you have to do this over numerous projects, you can imagine that these kinds of things can keep 20, 30, 40 people busy just in terms of doing the manual work. What we've been able to do is, again, look at data science opportunities where, using again ML and AI, we are able to automate so that the systems are able to automatically align the peaks, and then you're able to do an image analysis based on which you're able to translate that into a peak under the area. And based on our initial proof of concept, or some initial experiments, it seems like we're already able to get to 99% efficiency in terms of how the systems are able to predict, which is already a fairly significant opportunity that we believe it could open up. And then lastly, I want to talk about another persona, and now this is Antonio. He is in supply chain. And one of the complexities we see with this is usually the information on the supply side is not always matching with what is on the clinical side. And you know that in a situation like what we have with COVID, it's become extremely difficult to just keep track of these kinds of information. So, for example, if a hospital says, I have 50 kits, you never know if those 50 kits are adequate. Are they going to be adequate for the next 6 months? Are they going to need a resupply in another month? And what we do is, in the absence of those kinds of information, we drive blind. And because we are blind, we produce more material. And because we produce more, we also have a lot of wasted material. So what we are aspiring for here is, again, using IoT capabilities, for example, where we are able to get more real-time information from our sites, or we are able to use more analytics also to project how the site usage is, and accordingly translate that into how we should be planning our supply activities upstream. Now, the beauty of all of that is it not only reduces the time associated with getting what our patients need, but more importantly, we are able to be more agile. So, for example, in the future, if we want to go to more remote trials, or if we want to do something more engaged from a patient perspective, we are future ready because the data exists. It's just that we need to manage it in a fundamentally different way than we're doing today. Now, if I look at all of those examples that I mentioned to you, we are in different stages. Some of them in the early stages, some of them are in the later stages of actually doing the data science. So if you look in in this representation, data science is beautiful, right? It's sexy. It's the one thing you hear about everywhere. No matter which business you talk about, data science is everywhere. But of course, that is the top of the pyramid. And of course, that's where the knowledge is. But what we've realized, and I think all of you have mentioned at the poll as well, the challenge is not with data science. Yes, the technologies and capabilities are maturing, but very little attention has been paid to what happens below it, what lies below the the pyramid, and that is where in our world we generate a lot of data, right? Be it in transactional systems, the data part has been established in the industry for quite some time. The connectivity is something that we are just evolving right now in the area of IoT. But the biggest gap is in the middle, where we have how do you translate data to knowledge? And of course, first you need to convert that to information, meaning how do you get the metadata? How do you make it verified? You've heard it from Matthew. This becomes a very important element of how we can take what we have shown some examples previously in terms of data science, but most importantly, to be able to make it sustainable and scalable. The only way that can happen is by having such a fully integrated link from data to information to knowledge. And that's exactly where we've been partnering with Wolfgang and his team, with ZONTAL, to really help us verify the data and really build these data lakes so that we're able to do such seamless interface, for our predictive analytics capabilities. So with that, maybe Wolfgang, I'll hand it over to you to share how we've been doing with some examples.
Yeah, thank you. So let's have a look at what ingredients we need to build such a fair data platform at enterprise scale. So first, of course, we need some data. In order to make it findable, we need to understand what the context is. So we need to link the data to some reference or master data entities. Then in order to make it interoperable, we need to align the data to data models and standards that are reused across the data sets. Of course, as also Matthew mentioned, we need to consider security and compliance along the way. This will deliver us then verified data sets, and then we can access those data sets via a harmonized interface to have them in the end available for reuse in different types of applications. So we can integrate data sets in the operational workflow, data visualization tools, maybe for the citizen data scientist, and then more the heavy lifting data science for AI, ML. So let's look into a simple example of one of those data visualizations applications. So this application was developed by a data scientist at Novartis. So I think it was roughly an effort of 2 days, including the training, to really build this tool and make this available in the end to lab scientists so that they can run first analytics on top of the data without having any technical knowledge. So here we do some stability trending. We can select batches, conditions, and packaging information, and then get the data returned. And just to clarify some terminology for the ongoing discussions here, so these labels here, these are all concepts aligned to an ontology. Then we have all the reference data like the conditions. So we not only want to standardize on the ontology level, but we also want to try to harmonize on the data content level using these reference vocabularies. We have access to the master data, in this case, batch numbers that may come from SAP. And finally, then we have the actual results. So now one thing that we can do here with this tool is run a batch inspection to look into some outliers here to see where some batches out of spec. So how does a data scientist now approach this effort to really build and deploy tools to the lab scientists? So first step is he would go to the FAIR Data Hub to understand what do we have in the inventory. So what data is available. So Matthew calls this a data catalog. So here we see, for example, that we have stability and release data from the LIMS in the system. We can see the contextual metadata information that we can use for the data discovery. But what the data scientist now also finds is a link to the data model. So each data set in the hub is self-reporting, self-describing. So you can click on that link, and then we'll get access to the LIMS data model, which is then harmonized across all source systems. And in this case, we had 4 different LIMS data sources running together here into this harmonized representation. So this data model now contains 3 parts. So we have a core LIMS data model, and then we have 1 branch for batch release and 1 branch for stability. So we can even then, using this harmonized data model, run analytics across stability and release in a very simple harmonized fashion. Then the data scientists can zoom into the data models to understand, for example, here, what is the definition of analytical method that has been used. So this is the view now into the ontology. And the next step would be to understand also what kind of methods do we have. So these would be then used to build these filters in the dashboard application, for example, for the conditions. So here we have then another link tool that we can use then to look up all these method identifiers here in the end to understand what are the references and the data that we can reuse. So if we pull this all together, then from the data scientist perspective, he has now really a self-service environment where he is largely independent from anybody else to look into the data, build his own data science applications, or build and deploy applications for lab scientists. So he no longer needs to ask the business, for example, what data do we have, where is it, and how can I get access to that? So he doesn't need to bother IT to understand all the technical aspects, like what is the data format, how can I parse it, what is the data model, and then also, again, going back to the business to start interview, like what does this property mean in the data model, what do all the values mean, and who's an expert who can tell me more about that. So this typically creates a lengthy process that will run over multiple weeks, and now we've been able to condense this down to a 2 days exercise. So that was what I had prepared for the day. Thank you. Maybe Zaid, we want to take it on? Did we lose Zaid? Looks like it.
It does.
Thank you, Wolfgang.
Hello? So maybe then it's a gap filler.
But he'll be back with us shortly, I'm sure. Thank you very much for that. I'm going to just refer you straight to John Harmon's question. Excuse my doorbell going off. And John asks, are you making efforts to, and if so, how, move standardization, quality, curation, enrichment to the edge, point of acquisition, rather than to have to deal with what you get? I hope I've read that correctly. You get the idea.
Yes. Yes. Matthew, do you want to start?
Sorry, and I was going to, well, let's start with Matthew.
Yeah, okay. Yes is the answer. And that's in two ways. One, we are working with, well, three ways. One, we are working with core capabilities inside the company where we're generating large volumes of specific types of data, typically omics and imaging. And that's where we're trying to understand the upstream experimental design. And we've been working with those groups around metadata models to apply to data. We're working to improve our what we call data transfer standards, but you could argue those are metadata standards that we would use with third parties who are generating data on our behalf. And we've also got a master data management and reference data management program running, which initially is around establishing and agreeing the models for master data entities, but in the long run, that's to embed those into source systems. Now, when it comes to the kind of systems that you use to run live clinical trial data gathering, that can be, that's not something you can change overnight. So it takes a little bit longer in terms of updating those kind of systems, but that's the strategy. Where we own the data generation, we're standardizing it. Where we can define the standards with third parties, we're doing that. And where we can embed data quality at source in our master systems using master data management, we're doing that as well. There are three kind of parallel activities, but they all drive harmonization and standardization over time.
And Vijay, you
Excellent.
touched on that.
Can you hear me?
Yes.
Yep.
Okay, excellent. Sorry, we were having problems with the text. Sorry, Luke, you to carry on.
I was just going to say, Vijay, you did touch on this too, but do you want to expand on that? That we're talking about?
Sure. I mean,
yeah.
Sure. Within the CMC space, we are still in the early stages, but we foresee exactly a similar framework as Matthew has outlined, in that we have a lot of historical information, and for that, you obviously have to go through the painstaking process of automating as much as you can, and bringing it up to your data lake and verify it. But of course, what will be critical is that cannot be the mindset for the future, wherein we need to think about data governance, specific people who are assigned to these kinds of things, so that whenever new data is generated, we no longer are living with an exception, but really to say that is a default. We cannot just have smart scientists. We also need to have scientists who understand data, so that the data ownership goes hand in hand with the process understanding or the analytical understanding.
Excellent. Interesting question coming in on how you incentivize good data citizen behavior. So rather than getting IT to sort of fix the data discrepancies that we have, how do we empower the user or the generator of the data to operate as a good data citizen? Perhaps Wolfgang would like to start with that.
Yeah, I think key to this is that we build a data capture into the process. So it's really hard to ask people, after you have recorded all your data, please go somewhere and start to catalog it. There's no point in doing so. So we need to capture the data right the first time and use automation and data integration to avoid duplicate data entry, so that each property is only entered once as early as possible into the process, and all following process steps then would inherit that information.
Okay. Matthew, would you like to add to that?
I agree with all of that. And for a lot of our lab processes, we have got the integration of our lab automation with our IT and informatics systems. There's still work to be done, but in different parts of the pipeline are more sophisticated than others. But the principle about data quality at source, capturing your metadata as you generate the data, is absolutely critical. Additionally, I did mention it, we're looking at sort of the cultural aspects of where you've got a tightly integrated automated process and teams managing steps of the process as you go through an assembly line, if you like. And we have that, for example, in our screening for new leads, both large and small. What we tend to find is peer pressure. The downstream team complains to the upstream team that they haven't given them the information to do their job. So that's more of a stick approach, if you like, in that people get told off for not completing the metadata correctly. But there's also the carrot approach about, peer reward for being a good data citizen. And like I mentioned, we're looking at different ways of accentuating that. We've got a corporate reward system that's called Catalyze as an internal brand, and we've got an upcoming internal symposium, and we're going to be giving out points to people who are demonstrating good behaviors, for example. Seems cheesy, but actually one of the things that we've learned over many years is that certainly in a scientific community, peer acceptance and peer recognition are a very strong driver. So we're looking at that in addition to streamlining our automated processes.
Excellent. So Matthew, what do points mean then?
They mean prizes in the long run.
Yeah. Okay.
There's a catalogue that you get to choose from. It's a little bit like having once you've got enough points, you can get a variety of patio furniture and other things.
Oh, lovely. Okay.
It's a great big catalogue of stuff. If you get enough points, you can choose physical things.
Excellent. Excellent. Okay, great. So what I'd like to do now is to launch another poll. And Vijay, I'd like you to prepare yourself, because I'm going to ask you to comment on the results of the next poll. So Luke, can I ask you to launch the next poll? Okay, so here we are. At what stages of the drug discovery and development pipeline are you intending to use AI? So if I can ask our audience to ponder, but not to spend too much time on pondering, but to then answer the question and submit their answer to us. Excellent. So, Vijay, we have almost an equal, well, not quite actually. 29% of respondees are intending to use AI in target biology, and then lead generation, clinical trials slightly less on 19%, but almost an equal split between target biology and lead generation. How does that resonate with the work that you're doing in your organization?
Actually, resonates quite well, Zahid, because we seem to have a lot of enthusiasm in this topic. If you think there are a lot of opportunities now in terms of scanning through systematically all our databases and identifying the right target for the right molecule for the right indication. There is a lot you can do there. Or if you look at it in terms of translational biology, the explosion of opportunities related to imaging and all the advanced analytics are allowing us to look at potentially indications that we may not have even thought about when we certainly look at initial clinical trials. And lastly, when you look at the late phase, in terms of protocol generation, in terms of the execution, in terms of again finding interdependencies across comorbidities or potentially the patient adverse effects and all of these kinds of things. It just seems like there are opportunities at every part in the pipeline. And what it seems is, at least in our organization, we have great ideas being pursued in all in these areas. But we also have an opportunity where we have research and development coming together at routine time points and saying, hey, we have this, what you have, how can we learn from each other? Or the same also happens between the development organization and the commercial organization. So to me, it's a very fertile environment. There is a lot of cross-pollination of ideas, because if you think about it, imaging, maybe idea that is coming up on the research side may very well also be used on the development side or even in the commercial arena. So that's where I see all of these ideas being pursued in parallel.
Thank you. And sorry for putting you on the spot there. Given the results of that poll, Matthew, we seem to spend a lot of time talking about patient-centric patient-centric research.
Mhm.
How can AI enable that, or how can AI contribute to that?
That's a good question. I think the main way in which we are seeing it at the moment is it's twofold. One is that we're looking at models of response in patient data, and then trying to translate that back to novel targets. And the other is that we're looking at enhancing the patient data with additional other data that's been generated at the time. We actually capture a lot of data in our clinical trials that are not absolutely central to the null hypothesis we're testing in the trial, but they're very useful for broad understanding of disease biology or novel target biology. Later this year, we will enable the support for what I call extended primary use, and that's where you can reuse the data in the context of a family of trials, say for the same asset, a program of trials, if you like. And the idea there is that we look at using, for example, AI to predict non-invasive observations. Let's say it's an expression profile that's associated with potential for response or non-response to drug, and that can help you with your patient stratification.
Mm-hmm.
So we're both feeding backwards up the pipeline and feeding forwards downstream once all the policies and guardrails are in place to enable us to do that, which should be later this year, as I say.
Mm-hmm. Thank you. Maybe Wolfgang, you can take this question. How can AI ML help overcome the lack of predictivity from preclinical research models?
That's a good question, and I'm not sure if the right one to answer that. So I would say, if you cannot predict it as a human, it's hard to teach a machine to predict it, right? I refer to more the scientist here in the room. Maybe Matthew or PJ, you have an idea how to approach that problem.
Okay, all right. So Vijay, as a scientist, how would you approach this problem?
It is a very difficult question, and I think I would say exactly that AI and ML somehow have this unique expectation these days that they can change the world, and something that humanity cannot do by itself, a model can do. But the reality is the expectation is just unrealistic. If the question is more about, we have a lot of data, we simply cannot interlink and understand the interdependencies between data. That is, of course, where AI and ML can help. But if the data itself is not interconnected, obviously you cannot make conclusions out of it. So to me, it's both. There are certain opportunities, but I also think science also needs to evolve a little bit more, because that's not the state of science today.
Yes, absolutely. And Matthew, perhaps you can deal with this question. How do you determine what counts as complete metadata? Isn't there a chicken and egg with the level of analytics resolution here?
It is, yes. So what I was referring to earlier on was being able to compare the metadata on your data set to a standard that you need to achieve. And so what's complete metadata? If you could think about it in terms of the completion of all of the fields in your model, and that's a kind of a numerical address, but it may be that those aren't all the fields that you need for the particular question you're answering. Mm-hmm. It's a way of setting a quality flag. So you can say this study, at a high level, it's got all of the information in you'd expect for a study of high quality. Mm-hmm. Now, does that mean that you can answer the specific questions? It depends on how you drill down into the granularity, and when the more granular you get, maybe the less complete the metadata is, depending on the data capture. So I was thinking about it more when I was talking earlier as a way of setting a quality flag on the study level rather than the individual row data level. If you've got a metadata model and that's your standard, then you can compare it in terms of completeness. I think from an automatic point of view, comparing it in terms of accuracy is much harder, mm-hmm. because you can't tell automatically whether somebody's put an inconsistent value into that field. There are certain things you can do in terms of QC wrangling, but a lot of it is harder to do. We've been thinking about it in terms of ontological completeness. So the term in this column in your metadata, there's no reason it would never be there because of the type of field it is. But that's something we've looked at, but I don't think we've got an answer for it yet. It's a nice idea, but we haven't got very far with that.
Yeah. With another trope, we call this fit for purpose. So you're complete when you have described the whole universe on the microscopic down to the atomic level. That's not going to happen soon. Yes. So you need to understand what the use case is that you're trying to address.
Yeah,
absolutely.
Yeah. So we're going to be very busy then, if that's the scope. If we can project forward then, and we're all agreeing on the need to exploit AI and machine learning. There was a question early on about talent and skills, and maybe Wolfgang, you could start with this from your work with many of the leading pharmaceutical companies. Do we have the right balance of talent within R&D to enable us to reap the benefits that AI and ML can convey to us? And if not, what do we need to do about it?
Yeah. So one of the limitations that we currently see is how this talent that exists is being used. Mm-hmm. So we see that roughly 50% of the scientists' capacity goes into administrative tasks like reporting or data wrangling, curation, or whatever. So if those platforms help to offload some of this burden, maybe reduce it to 25% or so, that would be already huge. And as we have seen in this one very simple example today, it's already very efficient, like using a data scientist, use pre-curated fair data that is easy to consume, and then build his own tools. So without the need to talk to anybody from IT or so. So really the self-service community built around the combination of data scientists and lab scientists, things that we will see an increase in efficiency here.
Yeah. So Matthew, once you've got to the point where all your data is AI and ML ready, mm-hmm. and you're handing it over to the scientists, are they AI and ML ready?
So we recognized this as we were building out our strategy a couple of years ago, and I was actually a very strong proponent that we build in a learning and development capability. So as I sit in the data science and AI function in R&D, I would argue that having a strong central function is also a good idea. Mm. But we've deliberately put in a learning and development capability. We've got a data science academy that we've just recently launched, and we've 2 levels. One is the kind of general education and awareness around the potential for these kind of approaches, and the second is more hardcore expert training. Mm-hmm. Now that's our immediate response. We're making sure that we've got people who are trained in R and Python and Jupyter notebooks, and they're not afraid of an AWS command line, and they can start building models on their own. And then the next iteration beyond that is building out the systems based on top of the data sets that we've managed to ingest into the lake and bring together in a structured way. So to give more sort of citizen data scientist type interfaces. So the challenge there is that you need to perform a lot more pre-computation of the kind of relationships that people are going to want to query. Mm-hmm. If you're building your own and you've got access to the data files in an AI environment, and it takes you 2 or 3 days of your own inspiration to come up with an answer, that's probably fine. If you're a citizen data scientist and you have to wait 5 minutes for an answer, that's not fine. So we recognize that that's quite a challenge to go from where we are now, which is supporting and educating people to be experts, to providing a much broader access to people who may not have the hardcore AI skills, but they've got the subject matter expertise to take advantage of the data that we've brought together. So that's our next challenge after this.
Okay, well, thank you, Matthew. I'm going to leave us with that challenge in the air. And thank you again for your contributions. So thank you, Matthew Wooddock from AstraZeneca. Thank you, Vijay Raju from Novartis. And thank you, Wolfgang Kolsman from ZONTAL. So we will be continuing our digital dialogue session later on this month, and our next session will be on how structured and accessible research data can drive digital transformation in the lab. Please make a note of that in your diary. It's 30th of September, 1500 BST for an hour. So do save the date, and I hope very much to see some, if not all of you, at that session later this month. So thank you, our panelists and speakers, and thank you, our audience, for attending. attending and for giving us some great questions today. And I'm now going to close the webinar. Thank you.
Thank you all.
Thank you.
Bye.
Thank you.